Hey, you guys. Hi, everyone. I hope you all are safe and you all are healthy at your place. We are talking about one of the most interesting topic today in this Bridge Gap lecture, and we are talking about the topic osmosis. Well, you should wonder why pickles have too much of oil, why jams have too much of sugar. There should be some reason why. You will say, ma'am, for the taste. Do you eat oil of that pickles? Of course not. Then what is the need of that much of oil? You will say, ma'am, it is acting like a preservative. But do you think that is the only reason behind that? Well, no. Here comes the topic of osmosis. But before I teach you osmosis, first of all, you should know what exactly is passive. Passive. Passive means where the energy is not required. A very effortless process is known as passive. For example, if I talk about any perfume, if I'm spraying a perfume in one corner of the room, that perfume is spreading to all over your house, or incense sticks which your mother lights in the morning, that smell, that aroma, the fragrance that goes all over the house. Why? It is because of passive, a type of diffusion it is, but the process is passive. What is the meaning of passive? Passive means no energy expenditure. No energy expenditure. Where there is no need of energy, there is no need of money. What is our money of the body? What is the currency of our body? ATP. Where ATP, adenosine triphosphate, the currency of our cell, where this is not required, we call that process as passive process. And one of that process, passive, the type of it is osmosis. What exactly is osmosis? Osmosis, in very simple term, is movement of liquid. Movement of what? Movement of liquid. So liquid movement, or you can say solvent. Liquid movement or solvent, any solvent. From where to where it should move? Which moves from higher concentration to lower concentration. I would like to just make sure liquid concentration we will consider a universal liquid for now, and we know that is water, H2O. Right? So liquid movement, that is water, which is moving from where? From higher concentration, that means where the concentration will be more, to less concentration where the concentration will be less. Like how? Suppose this over here is a beaker. I am separating this beaker with a very specific membrane. Here there are 100 water molecules. Here there are 80 water molecules. If this is chamber A, this is chamber B, you tell me where will the movement take place? A to B or B to A? According to osmosis, it is higher to lower. That means it will be moving from A to B such that both the type, the concentration becomes 90-90. Correct or not? So liquid movement from higher to lower concentration. But then what is this specific thing I drew? I drew a membrane. Which membrane it is? It is semi-permeable membrane. What exactly is this semi-permeable membrane? Semi-permeable means that allowing only and only liquid molecule to cross through it. If there are certain solute molecules which are present in this particular chamber, these solute cannot cross the membrane. These solutes can't cross the membrane. Only and only liquids can cross the membrane. We say that membrane as semi-permeable membrane. So I will just go for a summary of entire osmosis. Then with some illustrations, we will understand the concept and some questions we will solve which are important for Olympiad point of view, which are normally asked in your school examination also, as well as college examinations also, and various other things. Okay. So what I said was osmosis is liquid movement or universal solvent water movement from higher to lower concentration via semi-permeable membrane, a kind of membrane which will not allow any solute molecules. Only solvent molecules can cross the membrane. Clear? Keep that in mind because now you will be answering my some of the questions. What I have done is I have taken situation A, where in this situation A, I am adding animal cell in all three. I am putting animal cell, where this is one animal cell in chamber A, this is one animal cell in chamber B, and this is one more animal cell in chamber C. What I'm saying is this over here outside is water. This outside condition over here is what? Let's say it is sugar or let's say salt solution. It can be anything, sugar solution or salt solution. And the third condition, again, this is over here is water. Clear till now? Now, next situation what I'm putting over here is, my question is, if an animal cell, let's say any part of the animal cell, let's say RBCs only, red blood cells, our blood cells of course, if that red blood cells, I am keeping it inside these solutions. You tell me in first condition, water will be moving inside the cell or water will be coming outside the cell? You tell me. Now think about what did I tell you. I told you that osmosis is liquid movement. Water is already there. I said universal solvent. That means we are talking about water movement. Movement of water. But from where? From higher to lower concentration. Where is higher? Outside or inside the cell? You will say, ma'am, outside the beaker is full of water. Cell is only this much. How much water it will carry? So what you can say in a situation, because water outside is more, it is high, and inside the water is less, the water will start to move inside. Now, if you're talking about animal cell, do animal cells have cell wall? They don't have cell wall. They have only cell membrane. Correct? So, if too much of water keeps on entering inside, imagine balloon in your mind. If I just keep pushing water inside the balloon, what will happen? It will burst, right? Similarly, this cell, if water keeps on going inside, it bursts. We say this type of situation where outside there is more water concentration, in the cell there is less water concentration as hypotonic solution. Hypotonic solution. Okay, and in the first situation what happened? The cell ultimately, first it got swollen and then it burst. Now let's come to second situation. In case of second situation, there is a sugar and salt solution. Sugar and salt solution. Water is not more outside. So where do you think the water will be more? Inside the cell. And osmosis say higher water concentration to lower. It doesn't say higher salt or sugar solution. It says higher water. So where is water more? Inside or outside the cell? Outside there is sugar and salt solution. So water is inside the cell. Hence in this condition, here it is more. Here it is less. So water will start to move outside. If water is going outside, what will happen? Cell will shrink. This type of solution where there is more concentration of other like sugar and solution or salt solution more outside and less inside. That means water is less outside. We say that kind of condition as hypertonic. Hypertonic solution. And what is ultimately happening to a cell over here? It is getting shrinked. Correct or not? Now in the third condition, you will say, ma'am, this is same as condition A. How it is different? Here I am putting certain criteria. Criteria is whatever the concentration of water looks outside. Let's say the concentration is, let's say in percentage, if I say 50%, inside also the water is 50%. Don't go for the size right now. Just look about my criteria which I am putting in this condition. If inside the cell also 50% water is there, outside the cell also 50% water is there, what will happen to the cell? You will say, ma'am, if everything is same, the condition will remain the same. No water is going inside, no water is coming outside, neither it is going shrink, neither it is going, neither it is swelling, right? That type of solution where the conditions are balanced, no movement is taking place, or movement is taking place but you cannot see any change in that, we say that as isotonic solution. And in this, what change do you see? Nothing. So no change. I hope you understood this concept, because if you understood this, the same to same criteria I am putting in the second condition, but with a small difference. In the first situation, I used what? Animal cell. In second situation, I am going for plant cell. Suppose I am going for plant cell. In plant cell, I added one plant cell in this, one in this, and one in this. 3 chambers, A, B, and C. In first condition, now you tell me what will happen. Osmosis is same, definition is same. The only criteria which is changed is our cell over here. In this also we can say that the first situation, the water is more outside, concentration of water is more. So again, this situation is hypotonic solution. In this also, the water will be entering the cell, but because the plant cell is having a cell wall. You think even if the water goes inside, the cell will burst? No, it's just like imagine this kind of situation where I am having a wooden box. Which box? Wooden box. Inside this wooden box, I am putting a balloon. I am adding a balloon inside this. And I am telling you, without opening this box, try to burst this balloon. You will say, ma'am, the wooden box is there. How can I burst the balloon? This is the same situation. In this condition also, here this box is what? Acting like a cell wall. It is acting like cell wall. And if cell wall is there, do you think that even if this balloon is inside, you can burst it if the box is closed? No, right? Similarly, as much water as it can enter, it can have the capability of holding it. Only that much water will be going inside. After that, this cell wall, this wooden box will not let more water to go inside, hence the cell will not burst. So in this, the cell, again, we can say that the cell will become swollen for sure, but bursting, no. Bursting will not take place in the plant cell. In second situation, when I'm saying that the plant cell is kept in the sugar solution, let's say sugar solution. Now you tell me, what will happen? You will say that, ma'am, in this condition, water will be going out of the cell. Here, instead of using the word shrink, which I used over here, of course, the type of solution is hypertonic only. This is hypertonic only. But here I'm not going to use the word shrink. The word which I'm going to use over here is plasmolysis. What is plasmolysis? Understand this. Suppose this over here is a plant cell. Let me just draw it in a simple way. This is cell wall. We know that the plant cells have cell wall. This over here is cell membrane. Correct? When the water is going out of a cell, that means the water is going out. what will happen in this condition is this cell wall is intact. You cannot do anything to wooden box. If the balloon inside is losing the air after, if you check after let's say 15 or 20 days, it will of course lose some amount of air. The balloon can shrink, but can that wooden box shrink? No, right? Similarly, when the cell is losing the water, this complete cell on the inner part will become flaccid. But anything happening to cell wall? No. This condition is known as plasmolysis. Plasmo, that means plasma membrane. This membrane is plasma membrane. I'm writing in short as PM. So plasma membrane is losing the water. The water has gone outside, but nothing happened to cell wall. Cell wall is still the way it was there. So plasma membrane, lysis, breakdown. Lysis means breakdown. Breakdown of plasma membrane is plasmolysis. Clear? So in case of plant cell, we don't use the word shrink. We use the word plasmolysis. In C situation, again the same thing I'm putting forward. 50% over here, 50% over here. Any change? No change. You know that. So in this situation, this type of solution is isotonic solution. And here, no change as such can be seen. Clear? So this completes the concept of osmosis. Why I am teaching you this? Because I have few questions to ask you which are very common in the entrance examination, in the Olympiad examinations, as well as normally in school exams also they ask these kind of questions. But very much common for entrance and the Olympiad exams. Let me just show you a GIF to keep you understand what exactly we did. You can see this. The first, the water is going inside. So it is getting swollen. In second, water going inside, water going outside, because the same water concentration is there. You cannot see any change in this as such. In third situation, if you see over here, the cell is shrinking. Why? Because it is losing the water. So it is a shrink condition. Okay, let's move towards some of the questions which I was trying to tell you. If I tell you that there is a bacterial cell, this is a bacterial cell. Okay, let's say this is a bacterial cell, and what I want to do is I want to kill this bacterial cell. Kill it. Okay, my question would be, which kind of condition should I keep this bacterial cell into? My options are A, hypotonic solution, B, hypertonic solution, C, isotonic solution, or D option, both A and B. Think about it and answer. Bacterial cell, it needs to be killed. In which kind of solution you should actually keep it? Hypotonic, hypertonic, isotonic, or both hypo and hyper, that is A and B. Let's just talk about bacterial cell a bit about. Number first question. Do you think it, before bacterial cell, you tell me, do you think anything can happen in isotonic? Let's go for that removal method where we will remove the option first which we think that it cannot be possible for sure, right? You tell me in isotonic a bacterial cell can be killed. Of course not, because in isotonic we never see any change because of concentration on both the sides in the cell and the where the cell is kept, it is both same. Correct? So in this option is something which we are definitely removing. It cannot be one. Now we have three options right now, A, B or D. Either it's both or either A or either B. Now if we are talking about bacterial cell, first of all you should know one of the most important thing. Bacterial cell, does it have cell wall or not? Almost 99% of bacteria except mycoplasms and everything, all the bacteria have cell wall. if bacteria have cell wall, even if you keep it in a hypotonic solution, you think it will burst. No, because it is the same situation. What we did was, for the plant cell, cell wall was there, the cell got swollen, but did it burst? No. Why? Because cell wall is protecting it. Similarly, as much water you can give this to bacterial cell, still the bacterial cell, nothing will happen, if even if too much of water can go inside, because cell wall is protecting it. If this option is gone, automatically this option is gone. So of course answer will be hypertonic. But try to understand the concept over here. Why hypertonic? Because if this is the bacterial cell kept in a concentrated solution, the bacterial cell will start to lose water. Can you survive without the water? Our 70% of body is water. Can you survive without water? No. Any cell without water is a dead cell. So in this, if bacteria is losing the water from the cell, that means that the cell is getting dehydrated. If cell is dehydrated, how it can survive? So hypertonic condition is basically the one which can kill the bacterial cell. And hence come to my first question of what I started the lecture with: why pickles have too much of oil? Why jams have too much of sugar? Pickles have oil. Jams have too much of sugars. Why? Because oil and pickles, both of them create the hypertonic environment. And when they create the hypertonic environment, any bacterial growth cannot take place. Even microorganisms, normally the growth of them cannot take place if the condition is hypertonic. In earlier times, now the medicines what we use are very advanced. What they do is they block the pathway of the bacteria so that they don't multiply in our body. But in the previous time, the medicines what we used to use, they have the same mechanism. Hypertonic environment. That medicine used to create such hypertonic environment around bacteria, the bacteria used to die, and hence the cure for bacterial disease. But now the technology is very advanced, so we go for broad spectrum medicines. We just block the pathway, we don't let the cell wall formation only take place. So that is the other part of the other part which we can continue later on. But hypertonic was the condition in the initial medicines also, because of which the bacteria didn't used to multiply. Hence in agricultural field also, if we want to kill weeds, the unwanted plants which are competing with the main plants, if we want to kill that also, for that also always salting is done. Salting, addition of salt. Why salt? Because salt is creating hypertonic environment. The weed is losing the water and the weeds ultimately die. Right? So some of the real life things what we use, but we don't know the scenario behind that. One such topic it was, that is osmosis. I hope you understand this lecture. In case of any doubts, please keep me posted and updated. Thank you very much.